First of all, I'd like to congratulate all the laureates. It's a distinctive privilege for me to be addressing such a distinguished audience. So today I'd like to talk to you a little bit about statistics and challenges in a data-driven scientific world. Galileo famously said that the laws of nature are expressed in the language of mathematics, and that's certainly true. And the development of mathematics has gone hand in hand with the development of physics for centuries. And this happy marriage continues to this day. And to some extent, that's what this prize recognizes as well. But I think in the 20th century, which was termed the Statistical Century by my colleague Brad Efron, we saw the eruption of a new science, namely statistical science, that extends considerably the range of quantitative reasoning, not only within the physical sciences, but to new sciences such as the life and medical sciences, the social sciences, economics, and so on. And so in all these fields, we have theories, and these theories can make predictions. And it is important to test the predictions against carefully collected data. Now, the thing about statistics is that sometimes these experiments, the experiments you do to test theories, do not yield a black and white answer or 0-1 answer, but rather you have to deal with uncertainty. And statistics is precisely the language that lets you address uncertainty and evaluate the strengths of a scientific theory against data. Now, this way of thinking has been extremely successful. I chose to emphasize an early success of statistical thinking. The Salk vaccine trial from 1954 aimed to eradicate polio in the United States. And what I'd like to emphasize here is statistics was not important only for analyzing data. It was, but also for designing the experiment itself. How we're going to compare children that receive the treatment, the vaccine, from those who do not receive the vaccine. And so this has been enormously successful, and so with enormous policy implications. And so now, of course, all these techniques have been perfected over a half a century, and now we have a rigorous process of clinical trials that bring drugs to markets, which, to the best of my knowledge, do not harm people, but rather help them. Fast forward to 2017, has something gone wrong? I'm sure you're all aware of a lot of scientific and media attention in an enormous problem that seems to plague scientific research at the moment, which is the lack of replicability. You see on my slides the cover of The Economist from a few years back. And so we see a lot of issues raised around the fact that science is the thing that should replicate itself, but it seems to replicate less and less. So perhaps you've heard about claims in the field of psychology and things like this, but it also affects the life sciences. And so there was a very influential paper published in Nature in 2012 by Bigley and Ellis that report that, for example, among others, Amgen, which is a biotech company located in Southern California, what they've done is they took 53 studies that they considered to be absolutely landmark in basic cancer science, and they set out to reproduce them. But to their surprise, they could only reproduce 6 of the findings. And so there's a German equivalent who tried to reproduce things with fairly similar results. So this is gaining enormous attention. Nature, Science, all the top journals are concerned with this. We see a lot of articles in the popular press about this, and that cannot certainly boost the public confidence in scientists and in science in general. So we have this crisis. Why is it happening? Well, there are many factors underlying the replicability crisis. For example, perhaps the publishing culture is at fault. Perhaps the granting agency culture is at fault that requires of researchers to claim extraordinary results before getting funding, and so on. And so, of course, all of these factors are way above my pay grade. But I think what I would like to emphasize is the statistical component of the replicability crisis. I think it is not a coincidence that we have problems with replicability precisely at the time where we're suddenly able to collect massive amounts of data. So as you all know, we're in the heart of the Silicon Valley. We collect more and more data, and everybody wants to use this trove of information to improve their business or their science. But I think what happens with this is that now we have large data sets, available prior to the collection, the formulation of scientific hypotheses. And so if you think about this for a second, what we've done is we've turned the scientific method upside down. Before we were formulating hypotheses, let's say, take the example of genomics, about a single gene, and we would carefully design experiments to test the function of this gene. But now, because of technological revolutions, we can measure the expression profiles of tens of thousands of genes at the same time, and we'd like to understand which one are in the pathway of some disease. So we've turned the scientific method upside down, and so now we have to develop new scientific and statistical tools to address this new paradigm where essentially we collect data before asking questions. In other words, we're going to look at a lot of data sets to formulate hypotheses and validate them on the fly. And there's nothing wrong with this, but we have to be careful with what we're doing. So what is an example of a purely data-driven question? Well, that's kind of what supported the initiative around the Human Genome Project. Here's an example of a data-driven problem. So I have a response Y. Let's say it's a phenotype, and it's whether I have some disease or not, or my amount of cholesterol, and so on. And now, because of technology, what I can do is I can measure thousands and thousands of things about me. For example, it has become extremely cheap to genotype an individual. And so I can measure lots of genetic information about you. So the problem that we face now as scientists is this: which genetic variations influence a trait, the risk of getting a disease? Okay, that's a question we need to answer. There are many questions in the data-driven science that we have like this. For example, and it was featured in the previous talk, we can monitor the gene expression profiles of many genes at the same time. We like to understand which one helped me determine the severity of a tumor. Or moving away from the life sciences, and something that I think, as you understand, in this AI-dominated world becomes more important, I might be interested in understanding which factors or variables determine whether a loan will be repaid or not. So in all of these examples, what we have is we have a response of interest that we would like to understand. We have lots of what statisticians call covariates, lots of variables potentially associated with this response, and I would like to select as many of these variables as possible without too many false positives, because I am not interested in going to a biologist and reporting a set of genetic mutations that has nothing to do with the problem I'm looking at. I do not want to run into the problem of reproducibility. So if I want to formalize the selection problem a little bit, then we can imagine that, for example, taking my genetic example, that we have a sample of people, and they come both with their phenotype and their genotype. And so I have an X and a Y, and I'm interested in predicting the Y. And so my task as a statistician is to predict whether a given genetic mutation will cause, or maybe cause is a big word, but will change the distribution of the response, the likelihood of me getting a disease. One thing we can formalize is, so I can measure millions of things. I can measure millions of mutations. I'm asking which of these mutations truly change the distribution of the response. So mathematically, you can think about this like this. I have a response, whether I have the disease or not. I have hundreds of thousands of mutations. I'm looking at one mutation, and I want to understand whether the distribution of the response changes whether I condition on this variable or not. That is, if you look at what's on the slide, is I want to understand whether this mutation provides information, additional information about the disease status beyond what the other variables already offer. Okay, so that's a tough problem, which is very different from what statisticians have looked at for many years. And so just to kind of explain a bit why this is delicate, we have now a lot of tools that can measure the strengths of dependence. We live in a computer age where tools are extremely complex and sophisticated, and so we can use them to actually tease out which variables are important and which are not. But they're so complex that we should use them as black box. And so to give you a sense of like how a data set might look like, here we have a phenomenon that we're looking at. We have 500 possible variables. And so I have this black box that gives scores to each variable. Hopefully a high score measures an important variable. And so I get a plot like this, right? So I get a lot of scores. And so now I can hold everything the same. The statistical model is the same, but I just change the sample. But I'm looking at the same phenomenon. And so I get a plot like this now. And if I change the sample, I get a plot like this. And if I change it more time, like this and like this. So you see, I have enormous variability that I need to account for. See, science used to be about distinguishing signal from noise for a single event. But now we have extremely noisy estimates about lots of things because we're looking everywhere at the same time. And so how are we going to do this? Because in... Oops, sorry. Okay. Okay. Okay, maybe we can. All right. So Siri seems to like what I say. Okay. Okay. And so we have only one experiment, and so we have to decide what to select. And so we have to select in the face of tremendous statistical variability that is poorly understood because we use this extremely complex black box. Okay. So one method to think about this is the method of knockoffs that I introduced with a former postdoc, Rena Barber. And the idea is fairly simple. It's fairly straightforward. Like for each variable, for each real genetic mutation, I may want to introduce a fake mutation. And the idea of introducing a fake mutation is to try to create some sort of a control group that I'm going to use as knockoffs. And so the idea of this method is to try to, oops, how do I go back? Okay, all right. Okay, so the idea of this method is to actually feed the black box with both the real variables and the fake, and to look at what the black box does on the fake data set to sort of infer what it's doing on the correct data set. So now one way to introduce a fake data set is by what statisticians will call a permutation method. That is, I could try to append to my genetic information a new genome that has no relationship with the response whatsoever because I could just impute the genome of someone else, hence breaking the relationship between the association between the phenotype and the response, and perhaps use someone else's genome as a control. But that method does not work. So what you see on the slide is a plot of feeding the black box with both the true genomes and genomes from random individuals and measuring the scores given by the black box. The red dots indicate variables that have truly predictive power, and the blue dots are those dots that do not have any relationship with the truth whatsoever. And so just focusing on the blue dots, the false discoveries, so to speak, I can see that by just using this little permutation method, this naive idea does not solve the problem. Okay, the distribution of the scores associated that I get by imputing a random genome does not look like what it needs to do. Instead, if I use these knockoffs, which are constructed in a very specific way, then we get a picture that looks like this. And so then I can see that the distribution of the fake scores seem to mimic the distribution of the null scores, those scores that have no relationship with the ground truth whatsoever. So it works, and not only it works, but it provably works. In fact, you can use these ideas to build a complete wrapper around a black box. That is, you can feed real data and these fake data that are carefully constructed such that you can look at what the black box does on both types of variables and select variables with guarantees of reproducibility. Now, there's a lot of stuff, of course, I'm not saying when I discuss these things, how you make them, how you select. Here, I'll just say one thing, is I want to kind of convince you that statistical reasoning is extremely important, and that also this requires a lot of sophisticated mathematical ideas. So the correct statistical reasoning and correct mathematical reasonings are crucially important. So one of the things in the last 2 minutes, do I still have 2 minutes? In the last 2 minutes, I would like to broaden the subject a little bit because I think I want to make a claim which is a bit grander than just being a pure scientist and addressing this issue of reproducibility, and talk about perhaps other contemporary issues. So obviously, science is about establishing causality. And clearly, if we knew how to do this from observational data, then I think this reproducibility problem would go away. But I think that by focusing on reproducibility, by paying attention to what's being truly selected and is it replicable in the face of enormous statistical variability, it is a step towards causality. And in fact, this can be made extremely precise in perhaps simpler models, where to actually go through the program I just outlined will actually discover causal variance. So this is a two-way street. But at the same time, we are in the heart of the Silicon Valley. And as you all know the power of AI systems, and everything is about prediction. And so what we've learned is that to be able to predict well, we don't need a mechanistic model. But one way is not to have a purely mechanistic model. Another is to have a model that pays no attention whatsoever to relationships that might be causal. So we can argue that if we keep an eye towards causality, by selecting important variables, we can perhaps improve those algorithms whose only goal is to predict by perhaps making them more robust and more fair. So I would like to hit on these last 2 points. The first is robustness. So you see a lot of articles, this isn't one published in the Proceedings of the National Academy of Science, that claim that, well, if you like curly fries, it's very helpful to predict your IQ. And that may be true. That may be true in this very particular sample. It might be true in that location in time and in space. And maybe it's true outside, I don't know. But the goal of predictive algorithm should not just be to be able to predict what's happening on this data set. But we want to be able to retain predictive power in circumstances that are a bit different. How about tomorrow? How about in a different city? How about in a slightly different population? We would like to be able to have predictive power which holds under varied circumstances. And that's really what causality is about. Causality is what remains invariant through different circumstances. So perhaps by focusing on causality and focusing on variables that are truly important, we can improve the robustness of predictive algorithms. My second point, which I think is extremely important, is the fairness of AI systems. Perhaps you're aware that Microsoft put out an AI robot named Tay that very quickly learned to become racist and sexist. AI systems work and use data that may contain many biases and that do not encode any sort of causal relationships. As a result, perhaps there was this famous story, this famous Harvard professor, Latanya Sweeney, she Googled herself. And as soon as she Googled herself, there was an ad saying, Latanya Sweeney arrested ? Asking her to run a background check. Similarly, when women look for jobs, they tend to be presented with lower paying jobs, reflecting biases and prejudice. These biases and prejudice are not causal. And so there is a great danger in learning from data to just reify those prejudice and biases and to make them the norm. And so we have to fight this back. And that by focusing on what's truly causal, my claim is that it may help address this problem and not reify prejudice and biases. So the next 10 years, well, we have lots of work on our plate. What I want to convince you is that learning from data is not trivial. It requires an intellect, and it requires a lot of mathematics and a lot of correct statistical reasoning, and these things are very subtle. Thank you very much.
Wonderful. Thank you, Emmanuel. Questions? Oh, right there in the back.
That was a great talk, thank you. I was wondering if you could speak on the dummy variable method that you created. Do you think it's feasible to create a metric that generates these variables in a more agnostic manner that you don't need a lot of domain expertise in GWAS, for example, and it would apply equally well to an RNA-seq data set? Or is it inherently going to require a real amount of tailoring to get these types of approaches to work?
Yeah, I think it's a very good question. At the moment, we know how to do this in a few specific applications, but it's true that we do not have a general recipe. So what is required of the method is to know something about the distribution of the covariates. Sometimes we have a lot of unlabeled data that we can use to learn this distribution, and that is a situation where the method could be deployed effectively. But when I have no idea about how the variables are jointly distributed, it's unclear a priori how I would do this thing. And so it's a question for open research.
One right here. Let's see, can we have a microphone right here? Please raise your arm again. Thanks.
Thank you. I'd like to know, do you think the lack of replicability is due to lack of good understanding of causality?
No, it's just like what happens.
I have a second question. This is the first part. Second part is, once I have a good understanding of causality, the Bayesian inference network would be a good method to really handle this uncertainty problems. Thank you.
No, the lack of replicability comes from the fact that people are given data sets, they do not really have a specific theories to test, and they're going to try a lot of different models until something seems to work out. That's where the lack of reproducibility comes in. The fact that we're looking at everything at the same time, and we're not taking into account the universe of scientific hypotheses that we're testing at the same time when we report findings. Right? We're not correcting for what I would call a selection bias. And that seems to be a major obstacle. As far as the second component of your question about Bayesian, yeah, it is possible that it's, of course, there are Bayesian methods that are advocated to I'm not exactly sure how it leaves with causality, but to try to adjust for the selection effect. And I'm not by any means trying to diminish their importance.
Question right here.
Yeah, thanks for the talk. One question was around the first point that you made, which is basically a restating of the maxim that correlation not equal to causation. And then my second question was around whether the irreproducibility is often the issue with the effort that goes into collecting the raw data in the first place, in that if there are variations in the way the raw data is collected, then of course it's going to show up in downstream processes.
I agree. Well, I teach undergraduates statistics, so I absolutely am fully on board with you. Causality is not correlations. That's what I tried to explain in the last part of the talk, where it seems that a lot of AI systems are just picking up correlations, but they may not have structural causal relationships. And so that's why I feel that they're perhaps reifying our biases and prejudice. And I think causality is a very hard thing to establish. And historically, we've relied on randomized double-blind experiments. But now I think it's important to try to make progress towards causality in the face of the enormous trove of observational data that we have. And to keep an eye towards causality when you select a model, when you feed a model, seems to me to be a good principle. To examine the variables that you've selected, see whether they reflect spurious associations or not, seem to me like a good principle.
Very good. We would have time for one more question. Oh, perhaps right here. Yeah.
In life sciences right now, or biomedical sciences, a lot of people are arguing that if you look at single cells, you'll reduce the background noise, and then you should be able to get at some of these issues. Do you think that's right, though? Because you can't really ever repeat the experiment, right?
Right. So
Yeah.
So of course, what we need to do is we need to be able to assess. Absolutely. I mean, science proceeds by following false leads sometimes. But what we don't want to do is we do not want to only follow false leads. And so to have a sense of the strengths of what you're looking at in the face of this variability is all I'm advocating. So a lot of modern statistical research is about the following problem now. It's essentially, I'm looking at thousands and thousands of genes, and I would like to be able to select genes that are possibly implicated in a pathway in such a way that I would give a set of genes to this distinguished audience. Well, maybe you would not waste more than 90%, waste more than 10% of your time. 90% of what I'm giving you is, in fact, related to the response, and 10% may not. We will never go around this, as you know, science is not black and white, and we'll never go around this uncertainty, but we can help reducing the amount of false leads that we're following and money wasted in following false leads.
Very good. Let's thank Emmanuel again.